So there are a number of practical considerations when using CRISPR-Cas9. So in terms of practical considerations for CRISPR single guide RNA design, there's a few of them. First of which is that, of course, as I mentioned earlier, Cas9, the SP-Cas9 anyways, has a recognition PAM site of NGG. And so if you decide to go with SP-Cas9, your target sequence has to have a proximal NGG in order to perform the edit. And so, at least in the human genome, that sequence motif, NGG, happens about every 8 to 12 base pairs. So there's lots of sites across the genome that can be edited. Some other considerations you have are that, again, designing good guide RNAs is more than just finding a sequence that's complementary to your target sequence. It's also about minimizing off-target sites. And so you typically, anytime you want to design a guide RNA to target, for example, the coding sequence of a gene, you typically want to search the rest of the genome to find whether there's any similar looking sequences that might also be targeted by the same guide RNA. And there's software that you can use to help you do that. Some other considerations are that, if your goal is to, for example, knock out a gene, you want to make sure that your edit will actually generate like a proper knockout. And so, for example, if you're knocking out like a regular coding gene, then you want to make sure, for example, you might want to tend to target early exons in the transcript to induce like nonsense mediated decay or something like this. Because if you target, for example, later exons, you might end up with a case where when the earlier exons get spliced together, you might actually have a partially functional gene or partially functional protein. And so hitting earlier exons might work a little better for knocking out genes. You also have the potential to use multiple guide RNAs. So you might, in order to make sure that you properly knock out a gene, you might make multiple targeted mutations to the same coding sequence to make sure that you knock it out completely. Something that's a little bit less obvious is considerations with respect to target sequence efficiency. And so what that means is that even if suppose you want to target a given gene with two different guide RNAs, even if both guide RNAs have no off-target effects and both guide RNAs have perfect complementarity to the same gene, they can make cuts with different levels of efficiency. And so basically cleavage efficiency can increase or decrease depending on basically the nucleotide composition of your target sequence. So for example, like a guide RNA that has a G at position 20 of your seed sequence, so just one base pair upstream from the PAM sequence, might be more efficient than guide RNAs that have a C at that same position, even if the whole guide RNA is a perfect match to their respective target sequencings or their respective target sequences. And so here's an example of some software that you can use to help you design good guide RNAs. And so something to keep in mind is that the total length of your guide RNA, which includes the tracer RNA, is about 100 bases. The actual recognition sequence that you're actually designing to be complementary to your target sequence is about 20 base pairs or 20 bases. And of that, there's a segment that's just immediately upstream of the PAM sequence, which is called the seed sequence, which typically needs to be, which has more stringent requirements in terms of exact sequence complementarity to your target sequence. And that seed sequence is about 12 bases. And so a lot of people have spent a lot of time trying to figure out, why do off-target effects happen? And, they try to develop software that tries to predict what sequences might yield strong off-target effects. And so people have come up with all kinds of like different rules about and different like scoring mechanisms for telling people, okay, this sequence is highly likely to be an off-target effect versus this other sequence. And so here's an example of some software that where you basically tell it, okay, here's a region on the genome that I would like to target. And what it does is it helps you design a guide RNA. So for example, here it gives you a candidate guide RNA, and it shows you that the guide RNA is perfectly complementary to the genome sequence, the target sequence that you specify. It shows you the relative location of the PAM sequence. And it also gives you, in this case, 3 potential off-target sites in the genome. And so they give you on the left-hand side the location or the scaffold where the off-target sequence might be. It gives you an alignment, so it shows you where all the mismatches of the off-target sites are with respect to the guide RNA. And so those are highlighted in red. And then it also summarizes for you, okay, how many mismatches were found in the seed versus in the total 20 base recognition sequence. And then for this particular software, it gives you what's called like a cutting frequency determination score, where here 1.0 means it's more likely to cut at this off-target site, and smaller numbers basically represent a lower probability of cutting at that site. And so software like this, and there's lots of software out there to do to help you design guide RNAs, but they kind of work in a similar principle. They basically just try to identify for you where potential off-target sites might be. So there exists a whole slew of protocols for detecting off-target mutations. And so one of them, for example, is what's called circle-seq. And so the idea here is that you can take your genomic DNA, you can shear it into fragments, and those fragments will be a collection of fragments that both have sites that Cas9 will cut with a given guide RNA and those that don't. So what you can do is you can take these fragments, you can circularize them, and then you can cut them with Cas9 plus your guide RNA of interest. And what's going to happen is you're going to be left with a bunch of fragments where the 5' and 3' ends basically correspond to where a double-stranded DNA break happened. And so with paired-end sequencing, you can get pairs of reads where each pair of read that corresponds to a single fragment, when you align them back to the genome, will tell you basically where double-stranded DNA breaks happen. And so basically the result of circle-seq will be a map of the genome where you'll see locations where double-stranded DNA breaks happened, and then you can basically use that to detect where off-target effects happened. And so we'll just kind of briefly mention alternative uses of CRISPR beyond genome editing. And so outside of genome editing, one of the most common applications of the CRISPR-Cas9 system are to activate or inactivate gene expression of a target gene. And so the idea is that if you take a Cas9 complex and you mutate the 2 nuclease domains so that they're inactivated, that means that Cas9 can no longer make the double-stranded DNA break. But what you can do is you can fuse that Cas9 nuclease null mutant to either a repressor or activator domain. And what that'll do is that if your guide RNA then brings this mutant Cas9 complex close to the transcription start site of a gene, then you can activate or repress the expression of that target gene. And so in the cases of activation, for example, you might typically design a guide RNA that brings that Cas9 complex to a region somewhere like 200 base pairs upstream of the TSS. Whereas in the case of repression, it's typically more effective to bring it closer to the TSS or even a little bit downstream, like 100 base pairs downstream of the TSS. And so that's the main approach that you can make CRISPR A or I happen. The other approach that you can use is that instead of fusing the Cas9 nuclease null mutant to an activator or repressor domain, what you can do is you can actually just modify the guide RNA so that it has like a binding site of an RNA binding protein. So those are typically what are called RNA aptamers. And so what will happen is that with that RNA aptamer that acts as a binding site for the RBPs, then basically an RBP that's been fused to either an activator or repressor can achieve the same activation or inactivation of the target gene. And so it's also pretty common to take Cas9 nuclease null mutants and attach, for example, epigenetic modifiers to them. And so we'll talk about epigenomics in a later lecture, but basically you can attach, for example, like histone methyltransferases or histone acetyltransferases to Cas9. And what that'll do is that you can use a guide RNA to guide your Cas9, say, acetyltransferase to a particular region of the genome. And then what that'll do is it'll essentially facilitate the addition of like acetyl groups or the removal of acetyl groups to neighboring histones. And in doing so, then you can kind of like, for example, turn on enhancers or turn off enhancers at different regions of the genome. And so you can also actually do, to some extent, imaging with Cas9. So for example, you can attach like fluorescent proteins to Cas9 and use guide RNAs to then essentially guide your fluorescent protein to specific regions of the genome to then do like either like live or fixed cell imaging. And so there's a ton of different things you can also do with CRISPR-Cas9, and so here's just a few other examples. So for example, you can modify your guide RNA such that not only does your guide RNA have a protospacer for recognizing and guiding Cas9 to a particular site, but you can add on like a piece of RNA that will be complementary to some other RNA. And in doing so, you can essentially guide some external RNA molecule into close proximity into some part of the genome. In a similar way, you can modify your guide RNA to contain RNA binding sites. So there's a certain class of factors called RNA binding proteins that bind specifically to recognition sequences on RNA. And so you can modify your guide RNA to have these binding sites, which then allow you to bring these RNA binding proteins into proximity to a particular region of the genome. And so finally, what you can also do is you can design like pairs of guide RNAs where one guide RNA targets one region of the genome, the second guide RNA targets a separate region of the genome, but those two guide RNAs have complementary sequence to them so that they also in some sense hybridize to each other. And in doing so, basically you can bring two different potentially distal regions of the genome in close proximity to each other in order to, for example, like you could easily imagine, again, kind of bringing an enhancer into close proximity to a promoter that you want to activate.